Greetings. Welcome to my YouTube channel. My name is Judith Constantine from MUHAS, Dar es Salaam, Tanzania. I facilitate pharmacognosy-related subjects at MUHAS. And today I'll be with you in part 2 of plant ergastic substances. Remember, we did part 1 of plant ergastic substances, and if you haven't, please get back to part 1, then we'll come to part 2 and part 3. You are welcome. We discussed a lot about these substances, and we ended up with protein bodies, where we saw the hyaluron grains, the crystalloids, and we were able to differentiate between the two. And we will continue with that because we left something, which is the ground mass. The ground mass of protein, how does it relate with the globoids? How does it relate with the crystalloids? And how does it relate with the hyaluron grains? The ground mass of protein or matrix refers to the general, often amorphous protein substance that fills the majority of the hyaluron grains. It is found in the hyaluron grain. So in some cases where we remember we discussed that some proteins may not have crystalloids, may not have globoids. In those cases, it's only the ground mass of protein that is within the hyaluron grains. So in many seeds, this protein mass encloses distinct internal structures like crystalloids and globoids. And when we talk of a ground mass protein, this is generally refers to the main non-crystalline protein material that fills the majority of the protein storage vacuole. That is the hyaluron grain. It fills the hyaluron grain. But globoids are discrete spherical inclusions found within the protein body. They are within the ground mass. It's like they float within the ground mass. And crystalloids, they possess a defined crystalline structure composed primarily of storage proteins that are suspended within the amorphous protein mass. So the crystalloids and the globoids are all suspended within the ground mass, which is found in hyaluron grain. So the ground mass is the background matrix in which other structures are embedded, if present anyway. If we have globoids and crystalloids, they are always within the ground mass. In essence, globoids and crystalloids are specific structured storage components that are embedded within the general non-structured protein ground mass of the plant seed's protein body. So we can get the picture that ground mass, globoids, crystalloids, all of them are incorporated within the hyaluron grain. So this is the hyaluron grain. It has the crystalloid, it has the globoid, and the background, the matrix, everything in here is the ground mass. So that's it in summary. The endosperm cells of nutmeg, anyone who knows nutmeg, each contain one large and several smaller hyaluron grains. I will share a certain article which has a lot of microscopic tissues for various medicinal plants, including this nutmeg. So hyaluron grains containing globoids are present in the endosperm and cotyledons of lean seeds. Some of hyaluron grains of the endosperm of fennel contain a minute cluster crystal of calcium oxalate. Others contain one or more globoids. So we shall see what these calcium oxalates are and how they actually look like when viewed under the microscope. Fats and fixed oils. From the biochemistry point of view, Fats and oils are glycerides or fatty acids which are widely distributed and occur in both vegetative and reproductive structures of plants. In contrast to proteins and carbohydrates, these provide more calories, and so they are a valuable reserve food materials, especially in hard situations. Just like humans, we depend more on carbs, but in hard situations, we depend on our body fats. They occur in seeds where they may replace the carbohydrates as a reserve material and are commonly associated with protein reserves. Under the microscope, fats and fixed oils in plant cells occur as small, highly refractive drops of globules. Highly refractive. They are visible as small, round, and clear organelles called lipid droplets, also known as oil bodies or oleosomes. Oil globules associated with hyaluron grains can be well seen in the cotyledon of linseed and colocynth, and in the endosperm of Nux vomica and umbelliferous fruits. At room temperature, reserve fats occur in solidy, frequently colored and crystalline masses which melt on warming, and feathery crystalline masses of fat occur in endosperm of nutmeg. This is the microscopic view of oil bodies. We say they are highly refractive droplets. All these are protein bodies or protein droplets. We are done with reserve materials. Now we move to secretory materials. Most of this part is meant for self-reading, but I will be specific to some parts. But they include enzymes, they include nectar, they include coloring matter, which is chlorophyll. We won't dig much into this. So the pigments are responsible for colors in leaves, flowers, fruits, and whatever. So you guys can go through these. They are just simple to understand. Excretory materials. I'll spend much time on these, especially on alkaloids and mineral crystals. You will read on by yourselves on some other issues. You can even comment, discuss in the comment section about these other materials that have been left out. So these excretory materials are ergastic substances that cells store or eliminate because they are no longer useful. So the plants excrete these so as to prevent toxicity and aid in defense, and they include tannins, alkaloids, resins, latex, oils, especially essential oils, mucilage, and mineral crystals. So plants lack complex excretory organs like us. We use skin maybe to remove things like sweat. We use our urinary tract to remove urine and the like, but plants deposit waste in leaves, in fruits and barks, all dead tissues, and sometimes they make them fall off as the means of defense. Examples of excretory materials I have pointed out, but in this part we are going to deal with alkaloids and mineral crystals. Alkaloids, these are nitrogenous waste substances found in various plant parts. The presence of these in plants correlates with toxicity, though they are not an exclusive indicator of toxicity. So, most well-known toxins are alkaloid, but not all alkaloids are toxic. We should keep this in mind. And some are essential nutrients, or some have beneficial properties, especially they are medicinally important. Other toxic substances apart from alkaloids may be glycosides. For example, if you have heard of cardiac glycosides, we have tannins, saponins, because of their biological activity, their mechanisms of action. Examples of medicinally important alkaloids. These are just very, very few examples among many medicinally important alkaloids. One is quinine and quinidine from Cinchona cerebra. They treat malaria. Another one is morphine and codeine from Papaver somniferum for pain management and for sedation activity. We have caffeine, the well-known CNS stimulant. This is mild. We have vinca alkaloids known as vincristin and vinplistin from Catharanthus roseus. They are used for cancer management. These are just very few examples among many. We have tannins. I'll say less about tannins, but these are non-nitrogenous phenol derivatives that are widely distributed in plants, and they occur in solution in the cell sap, often distinct vacuoles. The presence of tannins prevents pathogenic attack in plants, so they are there for defense purpose. We won't say much about tannins, but they are important in medicine due to their astringent properties. They are important in leather industry, they are important in dye industry, and they also have important antioxidant properties. Another excretory material is latex. This is usually a milk emulsion produced by complex secretory structures called laticifers in some families of angiosperms. Plants produce latex for defense against pathogenic microbes, so they seal the wounds to inhibit microbial growth. Most of the times, the source of latex is Havea brasiliensis. This is a plant from which we get natural rubber, and actually natural rubber is obtained after vulcanizing latex. So latex is the starting material for natural. This is how latex is collected by creating incisions on trunks. Their collection is, we call it pathological means of collecting. It is collected by creating incisions, then it is being processed to get natural rubber. Vulcanizing latex is the process of adding sulfur or other agents and heat to natural rubber latex to create strong, elastic, cross-linked polymer chains, transforming sticky, weak latex into durable, resilient products like gloves, tires, erasers, by preventing molecules from sliding apart when stretched. So, natural rubber is the end product of latex, is the finished product of latex. So the chemical bonding in natural rubber creates a 3D network that enhances strength, flexibility, heat resistance, and prevents stiffness. That's the difference between latex and natural rubber. We have the latex of papaya, which contains papain. That's an enzyme helpful for digestion of protein food material. And please go and check something interesting about this cow tree, Prosimum galactodendron, which produces the latex that is as much as cow milk, the one we consume and is being consumed by people in Venezuela, as the source, the substitute for milk. It has the contents and everything just like in milk. You will enjoy reading about that plant in your self-venture. Once something that makes latex being classified as excretory material, it is because it contains carbohydrates and proteins, but these components are not generally available for the plant metabolic use, and they, even if the plant is under starvation, under no circumstance will it use carbohydrates and proteins found in latex as rare food materials. So in the next part, that will be part 3, we will do crystals, where we shall see calcium oxalate crystals and calcium carbonate crystals. So don't miss part 3 of plant ergastic substances. Thank you and see you next time. Please remember to drop your comment, remember to subscribe, remember to like and share to whoever you think may benefit from this. And please, if you have something new, like a reference, everything you have that may be important in this, please put it in the comment section, or you can email me anytime you feel like asking or giving me something new. See you in the next session.